Okay, next presenter is Donovan. Time somehow. Oh. Go ahead.
Alrighty. Good afternoon, everyone. My name is Donovan McCrae, and today I will be presenting on my capstone project investigating the role of FTL0304 encoded sodium hydrogen antiporter and its role within the environmental adaptation of Francisella tularensis. So we've all seen an overview. It's basically the same thing for me. We'll talk about the history of the pathogen. We'll talk about what my specific research aim for this project exactly was, delve into the methods that I used to study this pathogen, and then talk about the results and their implications further on in this world. So, Francisella tularensis. It is a gram-negative Coccobacillus bacteria that is non-motile and aerobic. It was first identified in 1911, and its first human case was ever described in 1914. It is an obligate intracellular pathogen, meaning that it has to live within certain cells within the body, specifically macrophages, neutrophils, and dendritic cells, in order to survive and replicate. What's interesting about Francisella is that it's exceptionally hardy within its environment. It has a lot of characteristics that allow it to survive in wet conditions, in dry conditions, within dead animals, within low temperatures, and such. It's very hard for us to actually culture this bacteria. A case in point, we need very specific media in order to grow it. And even when we grow it on the specific media, it takes days to grow into a proper colony in order to study. Another interesting portion about this is the fact that Francisella is very ubiquitous within the environment. It is found literally everywhere, and it's found in literally most organisms within the United States, at least, which means it has no true reservoir, which means it's also incredibly dangerous for humans because it can be passed to us from anything, which is exactly why Francisella is a bioterrorism agent, specifically a Category A bioterrorism agent, which would put it in the same classification as anthrax, the plague, and botulism. Part of this we know is from bioweapons research that was conducted through the 1900s, specifically the Cold War, in which the US, during Operation White Coat, deliberately infected US personnel with the pathogen just to see how it worked. And then the Soviets, on the flip side, decided to make it more deadly and figure out whether or not we can use it to kill a lot of people. This has led to the assumption that Francisella is, quote, the most effective biological agent for its cost, which is slightly terrifying, but, you know, we live. The good thing about this research is that it generated the live vaccine strain, which is useful for us because we can study this bacteria in a lower BSL laboratory. But again, this pathogen is highly, highly lethal. It has an exceptionally low infection rate, as little as 10 colony-forming units, 10 bacterial cells can cause an infection within a human. And with that, the WHO once reported that as little as 50 kilograms of aerosolized Francisella would be enough to kill 10,000 people and incapacitate a further 250,000. So it's highly important that we understand exactly what this pathogen does to the human body. And for that, we know that Francisella tularensis causes the disease tularemia, and that tularemia presents itself in 3 clinical manifestations that are significant to us, at least. The first being ulcoglandular, which is caused by a bite by an animal or by a tick or something like that, and it causes a very noticeable ulcer and the swelling of lymph nodes and things like that, fever-like symptoms. The next is oropharyngeal, where the pathogen is introduced to the body through food and water. Very similar symptoms to ulcoglandular, but it's more noticeable that you have swelling of the maxillary lymph nodes, which you can see in the picture behind me. And then lastly, the one that is most interest for research is the pneumonic form of Francisella. It is rare, exceptionally rare, and exceptionally deadly. It causes about 60% mortality rate for those who are infected by it. And the problem with it is that it can be infected into you through inhalation. And it is also incredibly nonspecific. There's no real indication to tell you that you're infected with a pneumonic form of Francisella or a pneumonic form of another bacteria. So circling back to Francisella and the reason why it's able to survive within the macrophage, it has a preferential treatment towards the alveolar macrophages in and of itself. There are two proposed pathways in which this pathogen actually enters these macrophages, but as with most of the research with Francisella, it's not well described as of this presentation. One way is possible, it's using the opsonized pathway to bypass the alternative internalization of this pathogen. And another is through a non-opsonized pathway that may or may not use the mannose receptor. It's still unknown at this moment in time. But the result for both is that there is a massive increase in inflammatory cytokines, which then recruit more innate immune cells, which allows this pathogen to survive. The next and most critical part of the life cycle for Francisella is its ability to survive within the phagolysosome. As other people have stated, the phagolysosome is a very antimicrobial environment through low pHs, differences in nutrients and things like that. So we don't exactly know how it does this. We think that it possibly can do this through the exploitation of early and late endocytic pathways, but it can also bypass the pH drop of the phagolysosome in and of itself, either through blocking the VATPases or, which is important to our study, using the sodium hydrogen antiporter. And from here, I will begin replication rapidly. So now that we talked about the basics of Francisella, let's talk about the sodium hydrogen antiporter in and of itself. These are protein structures that are specifically used to control the concentrations of cations intracellularly, specifically sodium and hydrogen, or protons. These antiporters can be separated into three broad classes: the CPA superfamily, the IT superfamily, or the ion transporter superfamily, and the sodium transporting multiple resistance and pH superfamily, which is a mouthful in and of itself. As shown on the slide behind me, there is a lot of different bacteria that can use these sodium hydrogen antiporters, and they can be used for a lot of different things. Just kind of going over how these antiporters actually work, there's currently two proposed mechanisms for this. One is a pH sensor model in which at lower pH or acidic pHs, the abundance of hydrogen ions will cause them to bind to a specific center on the antiporter and cause them to be inactive. They're not capable of binding to sodium and transporting it in and out of the cell. As that pH rises, the affinity for the hydrogen proton to bind to the center drops, and now sodium can bind and be transported. This type of antiporter is very inactive and activated at certain times. Meanwhile, there's the competition model, where because both of these ions are both cations with a positive charge, they're in competition with one another in order to be transported in and out of the cell. And depending on the pH levels, they will alternate between transporting more sodium or more protons. This type of antiporter is always active. So for sodium hydrogen antiporters and their relationship to the virulence in multiple bacteria, it's a wide array because, again, these things are very ubiquitous within bacteria in and of itself. They can be used for things like salt resistance, pH homeostasis, osmoregulation, sporulation. It just depends on what this bacteria needs it for what time. And now for Francisella, we know that it encodes for 6 of these sodium hydrogen antiporters. And we don't know what 5 of those sodium hydrogen antiporters actually do. The one that we do know, FT0304, the one that we are looking into this study, is shown to have an effect on the survival within the macrophages and within the virulence in mice. So, since we know that FT0304 does this, we hypothesized that the sodium hydrogen antiporter encoded by FT0304 of Francisella tularensis is required to maintain a homeostatic balance of pH and osmolarity, whilst also providing an essential role in the intracellular survival and virulence. And so how did we study this? Basically, the bulk of the study was growth curves, growing this bacteria, its mutant, and the transcomplemented strain in comparison to the wild type in different conditions to see how these bacteria would respond. Specifically, we wanted to test a range of pHs from 4.3, which is the base pH of a phagolysosome, up to 8.0, which is very alkaline. Test different salinity levels from 250 millimolars of NaCl to 2 molars of NaCl. And then lastly, test how it reacts in the presence of a oxidative stressor, such as hydrogen peroxide at 1 molar. Then, because we wanted to check, since there are 5 other sodium hydrogen antiporters that this pathogen encodes for, we use QRT-PCR to check the overall expression of these other antiporters when that gene is knocked out. Because possibly, if there are 5 of these other antiporters, they're going to compensate at least some bit. And then lastly, we wanted to double check on the intramacrophage survivability of the pathogen. And we did this by infecting raw macrophages to determine the intracellular fitness in comparison to LVS and the transcomplemented strain. And now we can talk about the results. So what we first found is that this pathogen, when FTL-304 is knocked out, it has no effect on the relative survivability of Francisella within various pH conditions, all the way from the most acidic that you can get or see in a phagolysosome at 4.3, all the way up to the most alkaline at 8.0 that a macrophage would necessarily be at. There is very little difference, if any difference at all, between the growth rates of all 3 strains. Something similar we saw happened in the salt trials. Again, we tested a wide array of salt concentrations from the lowest at 250 millimolar, which is not that salty, to 2 molars of salt, which is incredibly salty. And again, there's not much, if at all, of a difference between the growth rates of our mutant, our transcomplemented strain, and our wild type. And sounding like a broken record at this point, in the presence of hydrogen peroxide, again, there was no difference in terms of whether or not the pathogen is able to survive in the presence of the oxidative stressor. So you can imagine that we're getting a little frustrated here. We're seeing that nothing's happening. It's all not showing any positive results. So we move on to the QRT-PCR and hope to see, and what we did see, is that there was an upregulation of all the other sodium hydrogen antiporters, such that there was at least a 5-fold upregulation of this. So this leads us to think, okay, it's being compensated. It's not necessary for in vitro survival for a lot of these things, but it might be functionally redundant if it was knocked out. But we go back to the in vivo analysis through our macrophages, and what we find is that there is a significant difference. There is a full, log change of at least half a log, maybe even a full log, in the amount of mutant that we were able to recover in comparison to the LVS and the transcomplemented strain. So again, what does this tell us? It tells us that possibly the deletion of FTL-0304 does not affect the in vitro growth characteristics of Francisella tularensis, since we saw that there was no effect on the growth in pH conditions of a wide range, salt conditions of a wide range, the presence of hydrogen peroxide. It's possible that the redundancy, the ability to have multiple encoded sodium hydrogen antiporters is able to compensate for this and allow the pathogen to survive for long periods of time. It would also explain that there's no alterations between the growth characteristics of these two. And also kind of circling back to the nature of sodium hydrogen antiporters, while yes, they can be used to combat oxidative stress, they're more likely to use for nutrition and more likely used to combat pHs and osmolarity over oxidative stress. So that might be a reason why the the hydrogen peroxide trial did not work. And then we can also say that FTL-304 does contribute to the intracellular survival of Francisella tularensis, again, as evidenced by the log change difference in the macrophage survival assay. So where exactly do we go from here? So it might be important or beneficial at least to look at a multi-gene knockout mutant for these sodium hydrogen antiporters, because again, as we saw in the QRT-PCR assay, there was an upregulation of the other sodium hydrogen antiporters, specifically FTL-0085. So maybe generating a double or a triple knockout mutant, we can see whether or not these sodium hydrogen antiporters are truly having an effect on the survivability of this pathogen in these conditions within the macrophage. Also, it might be beneficial to do more in vivo assays, because again, the differences for Francisella in the laboratory environment in comparison to the natural environment are pretty staggering. So it might be beneficial to study things like mouse infection assays, LD50s, ID50s, bacterial burden, and cytokine analysis, just to see what exactly is happening with this pathogen. And that is my presentation. That's